Hello everyone, welcome to the session of Animal Kingdom NEET Theory Part 1. Disclaimer, the images, text and other material used in our presentations are for educational purposes only. We respect the intellectual property rights of the original creators and owners. If you are the owner and have any concerns or issues, please contact us at sathee@iitk.ac.in and we shall remove the content immediately. Please note that the parts of the content may have been generated using the generative AI, which is totally experimental. Learning objective of today's session: to understand the fundamental principle and the criteria used for the classifying animals into the different groups based on their characteristics and the evolutionary relationship. Second, to study the unique features of the phylum Porifera, because today we are studying the phylum Porifera, so we will study about the unique feature of phylum Porifera, like including their cellular organization, body plan and central canal system. To explore the characteristics of the phylum Coelenterata, that is also called as Cnidaria, focusing on their radial symmetry, diploblastic organization and the specialized cells called cnidoblast cells. Then we will also focus on the phylum Ctenophora, that are commonly called as comb jellies, understanding their distinguishing features such as the comb plates and the biradial symmetry. And we will focus on characteristic of the phylum Platyhelminthes, that are commonly called as the flatworms, and that also include the triploblastic, acoelomate body plan and the bilateral symmetry. And if we see the index, then we will focus on the basis of classification and the classification of animal. In the basis of classification, we will mostly focus on the level of organization, symmetry, diploblastic and the triploblastic organization, coelom, segmentation and the notochord. And if we see the classification of animal, then we will look into the phylum Porifera, phylum Coelenterata, phylum Ctenophora and phylum Platyhelminthes. So the first thing we will discuss in today's session, that is about the basis of classification. How the fundamental features are used for the classifying animals and why there is a need for the classification of animals. So first of all, the first point here is multicellular eukaryotic animals are kept in the animal kingdom. What type of animals are put in the animal kingdom? Multicellular. The first point is the multicellular, because all the unicellular eukaryotic organisms were already kept in the kingdom Protista. And for the multicellular and eukaryotic animals, we are keeping all that in an animal kingdom. So today we are going to study about animal kingdom. And the different animals are present with the different structures and forms. So in the animal kingdom, you see so many different types of animals. They are having the different structures, different forms, right? And the number is a large number, that is in millions. So the millions of species of the animals have been described so far, and there is a need for the classification, and that becomes very important. Why so? Because to study the millions of animals and to retain what we have studied about, it's a very, very difficult task, right? We are not a machine. We have a limited memory, right? And memory also varies from man to man. So millions of species, how we will remember their name, their characteristic, their unique specific features? It will be a very tedious task for anyone to study millions of animals one by one. So we follow classification system to make our study easy and in a very systematic way. The classification also helps in assigning a systematic position to the newly described species. Suppose to a taxonomist or you as a taxonomist found any animal which has not been classified earlier. Now what you will do? You will note down some features that are fundamental features. The fundamental feature means how the organism look like, what type of digestive system, what type of circulatory system, whether they are having any symmetry, right? All these are the fundamental features that we see in every organism, right? And according to that, you will note down all those characteristics. There must be some very specific, unique feature that is found in that animal only, and that will be help you. These characteristics which you have noted down will help you to assign it to a particular phylum, because the characteristics which you have noted down will match to a particular phylum, and then you can place that organism in that phylum, right? So in spite of differences in the structure and the forms of the different animals, there are some fundamental features common to the various individual in relation to the arrangement of cells, right? So there are some fundamental features that are common in every organism. Like if you see that there is a need of digestive system, there is a need of reproductive system, right? There is a presence of symmetry, type of symmetry, right? So these are the fundamental features that are present in every organism, right? Though these features are used as the basis of the animal classification, and some of them are discussed here. So we will discuss about these fundamental features, and a part of that, there are some specific features that are found in that particular phylum only. So that thing also we are going to study about. So the first fundamental feature that we are going to study, that is about the levels of organization. All members of Animalia are multicellular, and all of them do not exhibit the same pattern of organization of cells, right? So members of Animalia, all the members of Animalia, what type of like the cellular pattern we see there in them? One common thing, that they are multicellular. Their body is made up of multicellular, right? But we will see what type of organization of cells. Multicellular, what type of organization? Whether they are having the cellular level of organization, whether they are forming the tissues, if there is any organ system is there, or any organ only there. So that thing we are going to study about, right? For example, in sponges, the cells are arranged as a loose cell aggregates. In the case of sponges, we see that cells are loosely arranged, not in a compact way, right? So loose cells arrangement do not form the tissue. Number one thing, they do not form tissue. For formation of tissue, the cells should be intact. There should be close contact between the cells, and then they can do one particular function. A group of cells performing one particular function, that is tissue. But here, all the cells are loosely arranged, and they are not, every cell is responsible for its own function, right? So they are exhibiting cellular level of organization. Some division of labor activities among the cells we see, like in the case of like a cell only, there is a nucleus, there is a mitochondria. Nucleus function is very specific, and the mitochondria production of the energy. So in the cells also we see some division of labors, right? So among the cells, there are number of cells and different types of cells. So every cell is responsible for a particular function. So that is why it is mentioned here that some division of labor or activities that we see in the case of cells. In coelenterates, the arrangement of cell is more complex. It means in the case of sponges, we have seen cellular level of organization, but in the case of coelenterate, the next very phylum, coelenterates, they are showing a complex arrangement of cells. Not like a loose aggregates of cells, they are showing the complex arrangement of cells. It means that the number of cells, they are compactly attached to each other, and they are forming tissue. So here what we see, tissue level of organization. So cell performing the same function and are arranged into the tissue, hence it is called as a tissue level of organization. So in the case of coelenterates, we are having tissue level of organization. Organ level of organization is exhibited by the members of Platyhelminthes. Now the next phylum, that is the after this Ctenophora, there we are having the first worm-like phylum, that is flatworms, Platyhelminthes, and they are having organ level of organization, because here what happened, different tissues unite together and form a organ. So one step ahead after the tissue level of organization that we see in the case of coelentrates. Platyhelminthes, organ level, right? Every organ perform a specific function. Now, in the case of animals like annelids, arthropods, mollusks, echinoderms, and the chordates, organs have associated to form a functional system. Now here there is a formation of system. There is not a single organ we are talking about. We are talking about a system. I'm taking an example of digestive system. In our body, like in case of human, the digestive system starts from the mouth and end to the anus. The whole alimentary canal is responsible for the digestion of the food. So we are having mouth, we are having pharynx, we are having esophagus, then comes the stomach, then small intestine, large intestine, rectum, and then the very next opening that is called as the anus after mouth. The two opening, complete digestive system. So you see the number of like organs are there. Not only that, there are certain digestive glands are also associated with the digestive system. Our liver, largest gland, associated with our digestive system. Pancreas also associated to our digestive system, all because all their secretions comes to the digestive tract, right? So here what you see, number of organs unite together and perform a function through a system, and that is called as digestive system, as we have taken example of here as a digestive system. So similarly, in the body you see there is a nervous system, there is a reproduction system, there is a circulatory system, muscular system, and also the skeletal system. So many systems, right? And every system perform a physiological function. So why this like upgradation we see, like we are passing from cells to tissue, tissue to organ, organ to organ system, because as we move from porifera to coelentrates, coelentrates to ctenophora, ctenophora to platyhelminthes, esculenthes, annelids, arthropoda, the organisms are becoming complex, and their complex body needs some upgradation of the system. If we are talking about the level of organization, there is an upgradation of like cells to tissue, tissue to organ, and organ to organ system level, right? So that is why we see the formation of the functional system that perform a particular physiological function. So this pattern is called as organ system level of organization. Such a pattern is called as organ system level of organization. Organ system in the different groups of animal exhibit a various pattern of complexities, right? So this organ system, we will see that in the different groups, you will see there is a pattern of complexities. What is that? Let us understand with an example. Now here we are taking an example of the digestive system only. Like such a digestive system in platyhelminthes has only single opening. They have a digestive system, but they are having only one opening outside of the body, and that serve as both mouth and anus. And that is why it is called as incomplete digestive system. incomplete digestive system. A complete digestive system has a two opening, that is the mouth and anus. So simple organism like platyhelminthes is not a very complex type of organism. So they are having incomplete type of digestive system. And one more important thing, that why they do not have such a digestive system, because they are actually absorbing the digested food material from their body surface only. So digestive system is actually absent, only one opening, that is called as a mouth in there, and that actually doing the function of both mouth and anus, right? But if you see the complex organism, you will see that they are this digestive tract, the digestive system, they are having the two opening, that is as a mouth and anus. So we see the complexities, the pattern in the organ system. Okay, one of the example here we have seen, like taking the example of a digestive system, incomplete digestive system, complete digestive system. Similarly, we are having the circulatory system as a two type of circulatory system we are having. Open type. What happens in that? We are having heart, and what is the function of heart? To pump blood so that it could reach to the every corner of the body, every organ of the body, right? And here the blood is pumped from the heart and it is poured all over the cells and tissue. It is poured all over the cells and tissue. It means that the organ and the cells are bathed in the blood, right? So this is the open type of circulatory system. Now the very next type is, in the case of circulatory system, is the closed type. Blood is circulated through a series of vessels. Means the blood is not directly poured over the cells or the tissue. It is transported via vessels, and we are having different diameters for the vessels, like the broader one, that is the arteries, then veins, and then capillaries. So the blood is circulating in a channel. It is not poured directly over the cells or the tissues. So here you can see open circulatory system that we see in the case of arthropods. And here you can see grasshopper or the locust is shown here. From the heart, the blood is pumped directly to the tissues, all over the tissues, right? So it is open type of circulatory system. Now here you can see closed circulatory system that we see in the case of Ferritima posthuma, that is the earthworm. The blood, they are having the hearts. Okay, from the hearts, the blood is circulated via arteries, right? So they are having the blood vessels. It is not open type, it is closed type. So closed circulation of blood is there. Symmetry. Animals can be categorized on the basis of their symmetry. Animals can be categorized as the basis of their symmetry. So let us understand what is symmetry. Sponges are mostly asymmetrical. If we take the sponges, then there is no symmetry in the sponges. So if you divide the organism through passing through its central axis through any plane, and if it divide the organism into two equal halves, left half, right half, and that is totally equal, mirror image to each other, then it is called as, it is a symmetrical organism. Symmetrical, it is showing a symmetry. And if it is only having one plane that divide the organism to left hand and right hand equally, then it is called bilateral symmetry. But there sometimes the organisms are like that you divide the organism but it do not divide organism equally. That we see in the case of sponges. So that is why they are asymmetrical. No symmetry is there. Any plane that pass through the center does not divide them into equal halves, and that is why it is called as asymmetrical. When any plane passing through the central axis of the body divide the organism into two identical halves. It actually divide equally. The organism is divided equally, left half and right half. Then, and it is passing through the many plane. Not only through one plane you can divide the organism into two equal halves, but there are many planes like this also, diagonally also, by this diagonal also. Then it is showing the radial type of symmetry. Coelentrates, ctenophores and echinoderms have this kind of body plan. Animals like annelids, arthropods, etc., where the body can be divided into identical left and right halves in only one plane, that exhibit the bilateral symmetry. So, in the case of the animals, like we are having the annelids, we are having arthropods, right? When you divide the organism from its central axis, it divides the organism into identical left and right halves, and that exhibit the bilateral symmetry. So here in the figure we can see out that this radial symmetry, this is one plane of axis that divide the organism equally. This is the another plane and this is the another plane. So number of planes are there that divide the organism equally, right half and left half, right? So this is the radial type of symmetry. Now there is only one axis in case of crab here. This is arthropodon, right? It divides the organism left half and right half equally. See the appendages here, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, right? So this is showing only one plane is there that divide the organism into left and right half equally. Then it is called as bilateral symmetry. Diploblastic and triploblastic organization. Animals in which the cells are arranged into two embryonic layers, an external ectoderm and an internal endoderm. It is called as diploblastic animals. For example, coelentrates. Two embryonic layers are there. Two embryonic layers. External one, that is called as ectoderm, and internal one, that is called as endoderm, right? And in between the ectoderm and endoderm, there is a jelly-like substance. It is not a cellular, it is a jelly-like substance that is called as mesoglea. So an undifferentiated layer, mesoglea. You cannot see here the derm term here. Like in the case of ectoderm, there is a derm term is coming out. In the case of endoderm also, there is a derm term. So what does this show? Dermis means cellular layer. Ecto means outer side, endo means inner side, right? So there is a mesoglea, that is not cellular layer. So these animals in which the developing embryos has a third germinal layer. Now there are some organisms in which the third germinal layer is found in between the ectoderm and endoderm, and that is called as mesoderm. Now here again you see, like ectoderm and endoderm, here also the derm term is coming out, right? So it is a cellular layer now. So in between they are present, like in between the ectoderm and endoderm, and that is why three germinal layers are there. That is why it is called as triploblastic animal, triploblastic. And from platyhelminthes to chordate, all animals are. This is the name of the phylum. From platyhelminthes to chordate phylum, the organism belong to it. They exhibit this triploblastic level of organization. So here we can see the figure also for the three germinal layer. This is the ectoderm and this is the endoderm. In between the ectoderm and endoderm, there is a mesoglea layer. Now in the case of triploblastic, we are having the ectoderm, we are having the endoderm. In between the ectoderm and endoderm, we are having mesoderm layer. So this is the difference, mesoglia and mesoderm. 2 germinal layer, diploblastic, 3 germinal layer, triploblastic. Now we will talk about the next fundamental feature, that is coelom, or that is also called as the body cavity. Presence or absence of a cavity between the body wall and the gut wall is a very important in classification. Presence or the absence of the cavity. If the cavity is present, then it is called as coelomate animal. If the cavity is not present, it is called acoelomate animal, right? So what cavity we are talking about? The cavity that is present between the that body wall and the gut wall. Body wall and the gut wall, that is called as a coelom or the body cavity. The body cavity which is lined by the mesoderm layer, that is called as coelom or the true coelom, means the inner lining of the body wall should be of mesoderm and the outer lining of the gut wall should be mesodermal in origin. So that is called as true coelom. Animal possessing coelom are called coelomate, and the animal that exhibit or that have true body cavity as also called as coelomate, like we see in the case of annelids, mollusca, arthropods, echinoderms, hemichordates and chordates, all are coelomates. These all are coelomates animal. They are having the true body cavity. In some animals, the body cavity is not lined by the mesoderm. It is not lined by the mesoderm. Instead, the mesoderm is present as a scattered pouches, right? Between the ectoderm and endoderm, this mesoderm layer is present, but in a very separate pouch, right? So that is called as pseudocoelom. That is called as pseudocoelom. So let us see what is coelom here. So this is the body wall, and you can see this layer is the inner lining of the body wall that is made up of mesoderm. This is the gut wall, and the outer layer of the gut wall is made up of mesoderm. So this is a true body cavity. Now here in the case of pseudocoelom, you see the mesoderms are present in between the ectoderm and endoderm, but in a separate pouches. So the lining of the body wall is not made up of mesoderm, and the body lining of the outer lining of the gut wall is also not made up of the mesoderm. So that is why you say that this is a false coelom or the pseudocoelom. No coelom is present here in this third figure. You can see no body cavity. Ectoderm, endoderm is present here, and it is totally compact. Mesoderm is layer. So no cavity is there. Acoelomate. So such a body cavity that is called as coelomate, and the animal possessing them, the animals in which this pseudocoelom is found, that is called as pseudocoelomates. And the one of the example is the escalbanthes. The animals in which the body cavity is totally absent, that are called as acoelomate. Acoelomate. For example, platyhelminthes. In the case of platyhelminthes, the body cavity is totally absent. Acoelomates. In earthworm, the body shows this pattern, and that is called as metameric segmentation. Metameric segmentation. From the outside also you can see there is a demarcation of the segmentation, and when you cut the organism from inside also there is a demarcation of the segmentation. There is a lining of the segmentation. You can say that from this to this there is one segment, and from this to this there is another segment, from outside as well as from the inside. Now in every segment there is a repetition of one or the other organ. That is called as metameric segmentation, and that we see in the case of earthworm. And this is a very unique feature of this earthworm or the Pheretima postum, or you can say in the case of annelids. Now we will talk about segmentation. So in some animals, the body is externally and internally divided into segments with a serial repetition of at least some organ. So that is the metameric segmentation. For example, in earthworm, the body shows this pattern called metameric segmentation, and the phenomena is known as metamerism. Notochord. Notochord is a mesodermally derived rod-like structure formed on the dorsal side during the embryonic development in some animals. So notochord is, the origin of this notochord should be from the mesoderm. And what is mesoderm? It is a rod-like structure that is present on the dorsal side of the organism during the embryonic development in some animals, right? So that is the notochord. And the animal in which the notochord are present, they are called as chordates. And in those which do not, it is not present, then it is called as non-chordates. Like from porifera to echinoderms, they are considered as a non-chordates. No notochord is found there. Now, animals are classified on the basis of some common fundamental features that are shown in the figure. And the important characteristic feature of the different phyla are described. So here you can see this figure, 4.4 figure in NCERT. So what fundamental features we have studied so far? We have studied about the level of organization, and there we saw cellular level of organization, and then there is a tissue, organ, and the organ system level of organization. So all this tissue, organ, and organ system level of organizations are kept here in one group, and the cellular level is kept in another group, right? So this is the level of organization. Next fundamental feature, symmetry. Here you see mostly asymmetrical. Then comes the radial, and then comes the bilateral symmetry. Now the tissue, organ, and organ system level of organization bearing organism, they exhibit either radial or bilateral. So it is divided into two parts. Now this cellular level, only one phylum is there, and that exhibit the mostly asymmetrical structure. So it is kept here, right? Now, if we talk about the next fundamental feature, then it is a body cavity or a coelom. Body cavity or coelom. Acoelomate. Acoelomate means no coelom is present there. No coelom is present there. And in the case of radial also, some organism that bears no coelom, right? In the case of bilateral, we see all the three different types of coelom, that is acoelomate, pseudocoelomate, and true coelomates, right? Now, by taking all these fundamental features, cellular level, mostly asymmetrical, acoelomate, so that forms the porifera phylum. Porifera phylum. Radial symmetry, radial symmetry, tissue level of organization, acoelomate. They are shown by Coelenterata, that is also called as Cnidaria, and Ctenophora. Now, bilateral symmetry, that is acoelomate, Platyhelminthes, no coelom, no body cavities seen here. Bilateral symmetry with false coelom or the pseudocoelom, only one phylum, that is Aschelminthes. Now, bilateral symmetry, and all are exhibiting true coelom. So from Annelida to Chordata, you can see that all are exhibiting the bilateral symmetry, right? Only this Echinodermata is here. Echinodermata. This exhibit the radial symmetry or bilateral symmetry depending upon the stages. Like it's a larval stage exhibit the bilateral symmetry, but the adult stage exhibit the radial symmetry. Very unique feature. Both the type of symmetry is seen in this Echinodermata, and in one organism only. Larval stage is showing some other symmetry, and the adult one is showing some other symmetry, right? So this should be remembered, and that is why it is like marked over here. Otherwise, all are showing the bilateral symmetry. Annelida, Arthropoda, Mollusca, Hemichordata and Chordata all are showing the bilateral symmetry, and also they are having the true coelom. Now we will study one by one each phylum. So the first phylum is here, phylum Porifera. Members of Porifera phylum are commonly known as sponges. Members of this phylum, they are commonly known as sponges. They are generally marine and mostly asymmetrical animal. They are marine, means they are found in sea and oceans. But some are freshwater also. Some are reported in freshwater environment. They are mostly asymmetrical animals. No symmetry reported. They are primitive multicellular animals and have the cellular level of organization. So, this is the first type of multicellular organism in which we see that they are still having the cellular level of organization. Cellular level of organization. No tissue formation has been taken place because it is having a very loose aggregates of cells. The cells are not compactly packed and they are not forming any tissue. Sponges have a water transport or the canal system. Sponges are having water transport or the canal system. Very unique feature of this phylum. So here you can see the canal system. If you see the outer body surface, it is smooth, but they are having very small pores here. You can see these are the small pores, like a structure, opening, small opening, right? And these are called as ostia. These are called as ostia. And from this ostia, the water comes inside. The water comes inside. And inside this, you can see there is a cavity-like structure, or you can say it is having a canal system. This is called as the spongocoel cavity. Spongocoel cavity, right? This is the spongocoel cavity. The water comes here, and from this is an big opening that is called as osculum. Osculum. The water comes from the ostia, goes to this spongocoel cavity, and from the osculum it comes out. If you see like the internal lining of the spongocoel cavity, you can see a very specific type of cell that is called as collar cell or the coinocytes. Coinocytes. Coinocytes, they are having collars and they are having flagella as well, right? So they take food material from this collar and it goes there, digests the food, and all the undigested food comes out from this part. So water enters through minute pores that is called as ostia in the body wall and into a central cavity. Spongocoel. What is the cavity called? Spongocoel. From where it goes through the osculum, and from the spongocoel cavity, the water goes out from the body through an opening, and that opening is quite large if you compare it with the ostia, and that is called as osculum. So the entry gate for the water is ostia and the exit gate for the water is osculum. The pathway of water transport is helpful in food gathering. Now this whole pathway means the entrance of water and the exit of water. So water not only coming only with water, it is having some food material with it. It is having dissolved oxygen in it, right? So the respiratory exchange, the oxygen is there. They are having the food materials. So that is captured by the cells, that is taken by the cells, right? So this water transport system is helping in capturing the food, respiratory exchange, and not only that, removal of waste also. The waste material is withdrawn in the spongocoel cavity, and from the osculum all the waste material passes out, right? Coinocytes or the collar cells line the spongocoel and the canals. All these coinocyte cells, they are very helpful in the producing a water current because they are having the flagella, na? So when this flagella moves in the collar cells, it produces the water current, and that helps water to continuously move out from the osculum and new water allowed to come inside. So this coinocytes or the collar cells are very, very important, and this is a very specific features of this phylum Porifera. Pore bearing organism, and from this pore bearing organism this Porifera term is coming out. Digestion is intracellular. The body is supported by the skeleton made up of the spicules or the spongin fibers. So they are having a very special type of cells that is called as spicule or the spongin fibers. Spicules or the spongin fibers, and that is helpful for making the skeleton of the body. Sexes are not separate, they are hermaphrodite. Sexes are not separate, they are hermaphrodite. That is, eggs and the sperms are produced by the same individuals. Eggs and the sperms are produced by the same individuals. So that is why the organism is considered as hermaphrodite. Sponges reproduce asexually by the fragmentation and sexually by the formation of gametes. So sometime what happened, there is a breakage of the organism breaks into 2-3 pieces, and here each piece now behaves like a new individual. Fragmentation, right? But sexually, for the sexual reproduction, they forms the gametes. There is a union of gametes, fertilization takes place, zygote formation takes place, and then comes the larval stages. Fertilization is internal, development is indirect. Indirect means there is a formation of larva, having a larval stage which is morphologically distinct from the adult. It doesn't look like adult. It is different look than the adulthood. Now, if we talk about the examples here, then they are having cycon, commonly called as a scypha. They are having spongilla. They are called as freshwater sponge, euspongia, bath sponge. So here you can see the figure of the different sponges. A cycon, B euspongia, and C is spongilla. Now we will talk about next phylum, that is called as coelentrates or Cnidaria. They are aquatic, mostly marine, sessile or free swimming, radially symmetrical animals. So they could be sessile. Sessile means they are attached to the substratum. They are attached to a particular substratum, or sometimes they are free living, free swimming. They can swim here and there in the water. What type of symmetry they exhibit? Radial symmetry. The name Cnidaria is derived from the nidoblast or the nidocytes. They are having a typical type of cells which is very specific to this phylum, and this phylum got its name from this type of cell only, nidoblast Cnidaria. So they are having a stinging capsule or nematocytes, and where they are present, such type of cells are present on the tentacles and all over their body surface. Nidoblasts are used for the anchorage, defense, and the capture of the prey. So this nidoblast cells, they are for the anchorage. They are for the anchorage. They are present on their body surface. They are present on the tentacles. So that helps them to anchorage on a substratum. Defense, they protect them from the enemies. When the enemies comes and attack on them, with the help of these nematoblast cells, they protect themselves. They fight with them. Even they can capture their prey with the help of these nidoblast cells, because they are having a very sting-like, a needle-like thing that can penetrate the organism and make the organism paralyzed. So with the help of tentacle, that paralyzed animals can be taken as a food. Cnidarians exhibit the tissue level of organization and they are diploblastic. So in the previous phylum, we have seen that they are having the cellular level of organization, but in the case of cnidarians or the coelenterata, they are having tissue level of organization. So here the many cells that are present, they are compactly arranged and all are performing one specific function, and that is why they are forming a tissue. Diploblastic only they are having the ectoderm and endoderm, and in between ecto and endo they are having the mesoglea type of layers. That is not cellular. Diploblastic. So here, this is the representation of a nidoblast cells. They are having a capsule-like thing, and inside that there is this sting-like thing. So when they are not outside, but when they see any danger, any organism is coming that could harm to this cnidarians, or if they want to capture a prey, they immediately take out this sting outside this capsule like thing, right? They have a central gastrovascular cavity. They are having a cavity inside their body, and that is called as gastrovascular cavity with a single opening, that is called as mouth on hypostome. Digestion is extracellular and intracellular. Some of the cnidarians, that is the corals, have a skeleton that is composed of calcium carbonate. Some cnidarians, not all, some cnidarians, like the one mentioned here, corals. Okay, we see in the deep sea, these corals. Now they are having a deposition of calcium carbonate that provides some sort of skeleton-like thing, and it becomes very hard, corals. Cnidarians exhibit the two basic body forms that is called as polyp and medusa. Very, very important point. They are having two body forms, polyp and medusa. The former is sessile, means the polyp is sessile. It is cylindrical in shape and attached to the substratum. And they are like hydra and adamsia. The polyp stages that we see in the case of hydra and adamsia, whereas the latter is umbrella shaped. The medusa is umbrella shaped, and they are free swimming like we see in the case of Aurelia or jellyfish. So two stages we see in the different organism, but in some cnidarians both the stages are seen in only one organism only, like here in the case of Obelia. Cnidarians which exhibit both the forms, that exhibit the alternation of generation, that once one stage is there and after that stage the second stage come, and again that gave rise to the first stage. So there we see the alternation of generation, that is from polyp to medusa and medusa to polyp. So that is called as metagenesis. Very, very important point, metagenesis. And metagenesis is seen in the organism that exhibit both polyp and medusa. So polyp produces by the asexual method medusa stage, and medusa produces polyp stage by the sexual method, by the sexual reproduction. So that is we see in the case of Obelia both the stages. So metagenesis can be seen in the case of Obelia. Now if we talk about the examples of coelentrates, then we are having Physalia, that is commonly called as Portuguese man of war, Adamsia, sea anemone, Penatula, sea pen, Gorgonia, sea fan, and Meandrina, that is called as brain corals. So here you can see the polyp and medusa stage. This is the polyp one and this is the medusa. Now in the case of polyp, you see this is a cylindrical like structure, okay, attached to the substratum. They are sessile and the mouth is toward upward. What about the medusa? Umbrella shape, free swimming, and the mouth is downward. So Aurelia, in the case of Aurelia, we see the medusa stage, In the case of Adamsia, we see the polyp stage. Next phylum is the Ctenophora. Ctenophores, commonly known as sea walnuts or the comb jellies, like the walnut structure. You have seen the walnut, right? So similarly, same like structure we see for the ctenophorans, and they are having the comb. They are also called as comb jellies because the bodies look like jelly, but they are having the comb like structure. They are exclusively marine. They are not found in the fresh water. All the organisms are reported to be found in marine environment. Radially symmetrical. Radially symmetrical. If you divide the organism from the center of axis, it can divide into two equal halves, left half and right half. But there are number of planes that exhibit the radial symmetry. Diploblastic organism with the tissue level of organization. Only two germinal layers, ectoderm and endoderm, and tissue level of organization we see there. Tissue level of organization. The body bears 8 external rows of the ciliated comb plates, which helps in the process of locomotion. So when you will see the figure of this, one of the example, Ctenoplana, so there you can see that they are having the 8 rows of ciliated comb plates. And that ciliated plates helps in the locomotion process. Digestion is both extracellular and intracellular. Bioluminescence, this is also one of very important feature. When the organism you see in the water, they exhibit light. In the dark also they exhibit light. So this is a unique feature of this organism of the phylum. Sexes are not separate. They are hermaphrodite. Both the sexes are present in the same individual. Reproduction takes place only by the sexual means. No asexual reproduction is found in the case of Ctenophora. Fertilization is external, means fertilization takes place outside the body, and it is indirect development, that is a formation of larval stages. Examples: Pleurobrachia and Ctenoplana. So here you can see the 8 rows of comb plates, this green lining, these are the comb plate structure that helps in the process of locomotion. Next phylum is the Platyhelminthes. They have dorso-ventrally flattened body. The body is very flat, dorsal surface, ventral surface, and they are called as flatworms. These are mostly endoparasite found in animals, including human beings. They are endoparasite. They are found as a parasite inside the human body or different animals. Flatworms are bilaterally symmetrical, triploblastic and acoelomate animal with the organ level of organization. They are showing the bilateral type of symmetry. Triploblastic. Triploblastic means 3 germinal layers we see in the case of Platyhelminthes. Acoelomate animals, no body cavity we see, and they exhibit the organ level of organization. Hooks and suckers are present in the parasitic form. They are having hook and suckers like structures that exhibit the parasitic forms. Some of them absorb nutrients from the host directly through their body surface, and that is why they do not have digestive system. Their whole body surface can absorb the digested food material as they are present inside the intestine. So in the intestine, when the complete digestion is there, all the nutrients are absorbed by these worms. Specialized cells called flame cells help in the process of osmoregulation and excretion. So they are having the very specialized cells that is called as flame cells that help in process of osmoregulation and process of excretion. Sexes are not separate. Fertilization is internal and the development is through many larval stages. Some members like planaria, they possesses high regeneration capacity. It means When the planaria is divided into 2-3 pieces, each piece will develop into a new planaria. So they are having a very high regeneration capacity. They can make up their lost body part. If we talk about the examples, then we are having Taenia solium as a tapeworm, Fasciola hepatica, liver fluke. So here we can see the two figures. One is of the tapeworm, that is Taenia solium, and liver fluke, Fasciola hepatica. Totally flattened structure you can see, dorsal surface and ventral surface. So this is all about today's session. Thank you very much for attending today's session.